All right. Good morning, everyone. I'll introduce our speaker for today, our last session of the year, and a perfect topic to kind of round out our last session and look towards the new year. So Marko Ludaic is a PhD candidate now in Sweden, in Stockholm, with Arne Olofsson, which for those in the CASP community, Arne and his group are key community members. They're the reason we had AlphaFold 3 as a baseline in CAS 16 and have done a lot of work uniting the community. So thanks to his group and everyone in his group. Um, Marko did his bachelor's degree in Belgrade, Serbia, and then moved on to do his master's in bioinformatics in Barcelona, Spain, and is now in Sweden completing his PhD. And if you haven't read his manuscript yet, it is a great manuscript to learn about the limitations and how we measure the accuracy of RNA structure prediction. And that is what he's going to be telling us all about today. So go ahead and take it over, Marco.
Thank you for the lovely introduction. Yes, so this was my first project, starting as a PhD candidate in Sweden and in Arne Elofsson's group, with the topic of limits of deep learning based RNA prediction methods. So I will start this talk by briefly introducing RNA, although I'm sure that everybody here already knows this. However, I think that's worth mentioning that for many years, RNA has only been known as this messenger molecule, the one that sends the information from DNA to protein, as its actual value was unknown and underestimated. RNA molecules are, however, highly dynamic macromolecules that adopt complex three-dimensional structures, essential for many cellular and viral processes. And lately, RNA has become recognized as a very potent molecule and valuable in the fields of molecular biology, pharmaceutical industry, biotechnologies. And different types of RNA, such as antisense oligonucleotides, small interfering RNAs, and microRNAs, have shown promising results as therapeutics. In 2025, predicting a single chain protein structure is mostly solved, very efficient and accurate, and most likely due to the enormous quantity of data available to train such models for predicting protein structure. On the other hand, modeling RNA structure remains much more challenging, and the main limitation from the computational point of view seems to be the limited availability of determined RNA structures, which is at least one order of magnitude less than for proteins. And secondly, RNA's conformational dynamicity complicates the application of deep learning models. Unlike proteins, RNAs are much more dynamic molecules, and they undergo huge conformational changes to obtain a shape that's required for binding to a molecular partner. So the aim of this project here was to answer the question that is, can we accurately predict RNA 3D structure? And to answer this, we used several state-of-the-art methods to model RNA. So this here is a timeline that shows many methods for predicting RNA secondary and tertiary structure. And of course, this is not the whole picture, as there are many more. We have here methods from Newsino algorithm, mFold, and Vienna fold from the time when I wasn't even born up until now, where we have AlphaFold and methods with similar network architecture that were developed in the recent years, and all or trying to predict the most accurate RNA structure. Methods that are shown in purple here and green are the ones that were used in this benchmark study. All the purple ones are able to predict only single-chain RNA structures, with exception of Chi1, which predicts RNA complexes as well. However, due to the token limit, so the sequence size limit, we only use it here for single-chain RNA prediction. And all green ones here are the ones that predict RNA, RNA, and protein RNA complexes, and those were used in the benchmark. The yellow arrow is when I started a PhD, kind of like a year ago, and then briefly after also this project. Additionally, I have to mention that we wanted to include also deep fold RNA and DR-fold, but decided to remove them from the main analyses due to two issues. So deep fold RNA was unable to generate full atom models for many structures, and this is something that one previous benchmark also reported. And on the other hand, the R-fold failed to predict structures whose sequence length was above around 200 nucleotides. So this would reduce our benchmark data set significantly, which is why we excluded those two methods from the analyses. To create a benchmark data set, we first collected RNA-containing PDB entries after September 30th, 2021, which was a training set cutoff date for the benchmark methods, and we also added CASP 16 RNA targets. Next, using the 80% sequence identity and CD-HEAT-est search, we filtered out sequences. We also removed sequences with ambiguous bases or those that were very, very small and with low complexity. And those filters were applied for both single chain RNAs and RNA complexes. Sequence identity, however, is not the only criteria that should be filtered out, because even though we might get rid of highly similar sequences, there can still be structural similarity between data from the benchmark and the data in the PDB. This is why we performed a structural similarity search using RNA-aligned TM score. And to do this, we calculated pairwise TM scores using RNA aligned between RNA chains in the benchmark data and RNA chains in the PDB, and took the TM score normalized by the length of the query structure. Then we took a TM score cutoff of 0.7 and filtered out those structures above this threshold. So this approach successfully identified highly similar matches, and below you can see 3 examples where the structure was initially a part of the benchmark data, that's in green, and it's structurally aligned with several other structures from PDB that were in the training set. So doing this filtered out such examples. Here, we illustrate how prediction accuracy depends on the presence of structurally similar examples in the training set. So we show this relationship both on the left before and on the right after applying sequence identity and structural similarity filtering using single chain RNA here. So the y-axis reports TM score RNA, which is computed between each predicted structure and its ground truth reference structure, while the x-axis shows TM score max, the highest structural similarity identified for each benchmarked RNA by searching against the entire training set, so taking the maximum value of the TM score. The figure below represents the same analyses broken down by RNA categories of TM score. Across all groups, the trend is clear. So the more similar benchmark structure is to any structure in the training set, the more accurately it tends to be predicted. After all the filtering, you can see that this regression line kind of flattens as we get rid of those that are highly similar to structures of the training set. And our final data set size for single RNA was 42. So now I will go on to the results section for single chain RNAs. For single chain RNAs, we used 4 different evaluation metrics, and those are TM score RNA, which is the TM score version optimized for RNA, and it's widely used for benchmark purposes. Then we used the interaction network fidelity, the second section here, which is an RNA-specific metric. It's basically Matthew's correlation coefficient between the interactions of the reference structure and that of the predicted structure. Then we also used the local distance difference test, LDDT, which is a superposition-free score that evaluates local distances, differences of all atoms in a model. And finally, we used global distance test as another evaluation metric for the global fold of RNA. So each box here on the plot corresponds to one method, and each method is colored differently, and they're placed in decreasing order by mean TM score. And our results here suggest that AlphaFold is the best performing method, followed closely by bolts. Although when we compared those two, there was actually no significant difference between them. Next, we wanted to identify models that are of a good quality, and for this, we put a threshold for each of the four metrics. So success rates achieved by all methods are defined as the fraction of acceptable models with a minimum GDTS and TM score of 0.45 and LDDT and INF score of 0.75. And these thresholds are the ones used in the latest CASP evaluation of nucleic acids. So taking these thresholds and saying that, for example, we will accept the model as a good quality model if only 2 out of 4, 3 out of 4, or 4 out of 4, so all of the metrics are under the thresholds, leads to a total of only 12 good models for bolts and 11 for AlphaFold. So not a lot of models at all. When examining success rates using those thresholds, we observed that among the two global fold metrics, so the GDT-TS and TM score, GDT-TS yields substantially higher success rates than TM score across all methods. In other words, many models that are classified as correct by GDT-TS are not considered successful by TM score. So to better understand this discrepancy, we examined structures with high GDT-TS but low TM score. And three most pronounced examples are shown right here. So these structures also exhibit high INF values indicating well-captured base pairing, stacking, and local interactions. Therefore, those low TM scores are not necessarily reflective of a poor structural quality, but rather a limitation of TM score applied to short RNA chains. And this behavior comes from normalization by the length-dependent scaling factor, this one here, D0, which is used in the TM score formulation. So for short RNAs that are less than around 40 nucleotides, as we found, the scaling factor becomes so small that even minor structural deviations lead to very, very low TM scores. While TM score for proteins proposes a minimum D0 of 0.5, RNA implementations have allowed for even smaller minimum values. So it's sort of underestimating the accuracy of correctly folded short RNAs. And the previous benchmark study also noted this issue and addressed it by excluding all RNAs shorter than 60 nucleotides from the benchmark. However, we consider that this filtering is undesirable for several reasons, as there are biologically important RNAs that are shorter and structured, and excluding them removes a meaningful part of the structural space. And also because the methods evaluated in this benchmark were trained on the entire PDB, which also includes shorter RNAs, excluding those from the benchmark would prevent a fair assessment of model performance. So we decided to keep shorter RNAs here as well and continue with the TM score RNA as a widely used metric. Next, we divided our RNAs into four different categories based on their structures, and those are L-shaped RNAs, RNAs with many, many loops, complex RNAs, which have very intricate motifs and are usually much longer, and finally, quadruplex structures. So here we show that the distribution of TM scores for each category, and you can clearly see that RNAs with simple loops and L-shaped RNAs, like those two here, which are also well represented in the PDB database, they are modeled more accurately compared to complex RNAs like this one here in purple and quadruplex RNAs. The models on the right that you see are the best models within each of these four categories. Here on this plot on the left, we show a correlation between correlation of TM scores between balls and AlphaFold 3. And we observed that there is agreement where those models that are low scoring in one method, they're also predicted less accurately with the other method. And I also wanted to depict this quadruplex group here because I found that interesting that these very small RNAs were the worst predictions. So here at the very bottom in orange, and these structures here. And if you take a look at these are all AlphaFold predicted structures in orange, you can see that AlphaFold can actually recognize the overall quadruplex fold, but it's not able to predict the exact atom coordinates for these structures. Briefly here, I show TM score RNA as a function of MSA depth in a logarithmic scale for all of the methods that use MSAs for single chain prediction. And it's no surprise, I assume that there is a strong correlation observed for all methods here. The last section here would be our evaluation of RNA, RNA, and RNA protein complexes. And for that, we used two metrics. One of them is US-aligned team score that was expanded to allow evaluation of RNA complexes, as well as docu score that measures how accurately predicted is the interface between two chains. Docu was also used as a measure in the recent CASP evaluation of nucleic acids. So on the left, we have results, violin plots for the TM score, and on the right are results for the total docu score. The total docu score meaning that it's averaged across all the interface docu scores for the whole complex. So looking at these violin plots here, we can see that, again, the top two methods for RNA complex prediction are AlphaFold and Boltz, based on both TM score and docu score. In addition to this, we also calculated interface docu scores for each method to compare if there is any difference between modeled RNA, RNA, and RNA protein interfaces. And we found out that there is no significant difference between modeling these two. The scores are actually very similar in both cases. And the scatter plot on the left, on the right, the scatter plot on the right shows the correlation of TM scores between the top two methods, AlphaFold and Boltz. And you can see for a couple of methods, AlphaFold was actually better here. We have noticed that for all four methods, same RNA complexes were always the top ones. So we decided to take a closer look. And as you can see, both of these two top modeled RNA, RNA, and RNA protein interfaces, but also the overall global fold is modeled correctly. So they both have something in common, and that is that this RNA in both of these cases is actually double helical RNA, which is likely benefiting from its base pair complementarity, and therefore it's easier to predict. On the other hand, protein structure has surely been seen by the deep learning method during training, so the overall fold is accurately modeled as well. In addition, we also wanted to observe a correlation between the TM score and the interface docu scores. And so for each method, so that's why there's four scatter plots here. And if you take a closer look, there's a bunch of models where we have very, very low interface docu score, but we have a high TM score. So we started questioning how does such model look like and why do we have such a big difference between the team score and the docu score, even though they measure slightly different things. We would assume that if the global fold is predicted accurately, that the interface would also be predicted more accurately. And what actually happened in many models here is this situation. So these are the two models that have the highest difference between docu score and team score. This one is also animation, but it's unmoving. Maybe I can, yeah, okay. You just have to imagine that it's moving as well. I think it's clear to see anyways. So you can see that there in green, there's an RNA from the reference ground truth structure, and in red is the modeled RNA. And you can clearly see that the protein in both cases is actually modeled correctly. So it's the RNA that it's misplaced, which is why the interface docu for RNA protein is very, very low. However, TM score is high because most of the scoring goes to the protein part anyways, while RNA is at a totally wrong binding site. And this happened in both of these examples here. Next, we just wanted to also take a look at how reliable are predicted TM and IPTM confidence scores. And in these scatter plots on the y-axis, we take a look at the interface docu score. And then on the x-axis, it's IPTM score, which should tell us what does AlphaFold or any method think how good is the prediction. And you can see that for many models that are for many interfaces that are between RNA and protein, IPTM tends to get higher compared to RNA and RNA interfaces. And we believe this is because because of the protein part, which AlphaFold is able to confidently model the protein part, which is kind of why if there's a protein, the IPTM score and the interface, the confidence goes up and becomes much higher. So finally, to just briefly conclude, deep learning has advanced RNA structure prediction, but still has a couple of limitations, especially when it comes to modeling RNA complexes and binding sites. Because only 1/3 of the models actually achieve high accuracy. And accurate predictions mostly occur for RNA folds that are already well represented in the database, which is why this limited RNA structural diversity actually restrict generalization to novel RNAs. And current methods struggle to identify correct models reliably. So future progress, we can say that depends on having more and more and more experimental RNA structures, better accuracy metrics, and continued machine learning and deep learning innovation. Thank you for your attention, and I'm happy to answer any questions.
All right, thank you very much. So if everyone can either raise their hand for questions or type it in the chat and one of us will read it out. I can get us started. I have a question about the two examples you had for DocQ versus TM score, those RNA protein complexes. I'm curious, you said it was like a docking problem. Was the RNA fold reasonably close to the native and it was just how it was docked in that was wrong, or is the RNA fold also completely wrong?
Yes, so in those particular examples, RNA fold was actually predicted accurately. So it is actually the docking problem. So in the most examples, it is the docking problem. It's a wrong binding site, basically. And then also there was a couple of other examples where if the protein is symmetrical, a lot of the times the binding will be on the RNA will be bound to the opposite side, but it will be modeled correctly by itself. Yeah.
Interesting. Cool. All right. It looks like Elise White has a question. Do you want to unmute yourself,
Yep.
Elise?
Yeah. So it's funny, Rachel. I was just thinking about this, and forgive me, this might be a naive question, but I'm wondering how you know that predicted binding pose is wrong. Is it possible that that's just one possible binding register that the RNA might explore, even just transiently? Um, like I know it's not what's captured in the experimental structure, but nonetheless.
Yeah, I mean, from this perspective, when we evaluate the deep learning methods, we have to use ground truth structures, so we need a reference. It is possible that maybe this protein is able to bind another molecule at that binding site, and that's why this happened. But for this particular RNA, I would say it was supposed to bound on another site, but it didn't.
I see. Yeah. Yeah. I understand. Yeah.
Mhm.
All right. Anton, do you want to unmute yourself?
Hi, Marco. Thank you for the great talk. I have a quick question about multiple sequence alignments. I noticed that you said that the better the alignments were, the better the performance of the methods was as well. And I'm wondering if you could talk a little bit about what these multiple sequence alignments were, how they were constructed, and also if you think it would be fair to say that improvements in the multiple sequence alignments methods as well might lead to better 3D structure predictions.
Mhm. I mean, this is something that well, for proteins, this has been analyzed already before, and we do know that the deeper the MSA gets, we can get better predictions because we can extract better evolutionary signals. Um, and our results here doesn't suggest that we get much better accuracy. It's not as there is a strong correlation, but it's not out of the ordinary. And our MSAs were constructed using our MSA. It's a package that construct construct constructs MSAs for RNA using RNA Central database, our FAM database, and NT database, nucleotide database. Um, and yeah, sorry, I don't know if there was more to this question here.
No, you answered it. Thank you so much.
Yeah, thank you.
Right. I have one last question, I guess, then to close it up. Um, for TM score, so the size dependence of TM score has been, something that a lot of people have
Yeah.
thought about, noticed, and nothing has really been done about it. So I guess what are your proposals? How do we, still focus on small things? What are the methods you would try to correct these type of problems? And how would we even decide on what's potentially a better kind of size dependence parameter? How do we go? How do we
Uh,
improve this? I guess is the question.
yeah, so that was we also discussed this a lot after we've decided to use TM score RNA. Well, RNA Align actually adapted TM score for RNA, and they say so it's length dependent. The problem is that the scaling factor becomes very, very small for very small RNAs. So maybe just putting a threshold for D0 for the scaling factor, that's not going to affect the TM score further. Um, could could be something that that can maybe be done, but also just what we decided to do here is that, of course, use other evaluation metrics that not only rely on team score, precisely because of this problem. Some other authors, they suggest that we should just exclude short RNAs. But I think that using a combination of evaluation metrics, I think that it's the best case here.
All right, then thank you very much. If there's no further questions, we will stop the seminar for now. Let's thank our speaker, Marko, one more time. Zoom version of things. Everyone, we will see you. We're going to be taking a little bit of a break for a month-ish, and we'll return Jan 8th for a talk by Marta Sachwich, I always say her name wrong. And then we'll continue bi-weekly from there. So we will see you guys all after the new year. All right. Thank you again, Marko.
Thank you so much.